They found her in the driest place on Earth, the Atacama Desert, Chile, a land where rain hasn't touched soil in over 400 years. It's a place where time forgets to rot the dead. But what they uncovered in 2003 didn't look human. Not quite. Just 6 inches long. 10 ribs, not 12. A skull stretched like a cone. Bone plates fused too early. Fingers too thin. A face too sharp. Like it was sculpted for zero gravity. They called her Atta. And the world lit up with theories. Alien child. Experimental clone. Hoax. TV crews flew in. UFO researchers swore it was proof. Even seasoned pathologists couldn't explain it. The desert had preserved her too well. Too perfect. For years, no one could prove what Atta was. Until 2018, when 2 scientists at Stanford University, Dr. Gary Nolan and Dr. Atul Butte, finally sequenced her entire genome. The results would be published in Genome Research. Cost: just $1,000. Impact: priceless. Atta wasn't from another planet, but she was genetically unprecedented. She was human, a female fetus, just 15 to 20 weeks old. But her bones, aged like those of a 6-year-old child. A body that defied biology. Her DNA revealed 7 gene mutations. 4 of them, completely unknown to science. Mutations that disrupted bone ossification, collagen pathways, the very scaffolding that holds a body together. Among them, genes like COL1A1, COL2A1, FLNB, associated with skeletal dysplasia, brittle bone disease, and dwarfism. But not in this combination. Not this severe. This wasn't one disease. It was a genetic catastrophe. And yet, the fetus had formed, developed, been born and buried. Atta's existence forces a question biology wasn't ready to answer. How did one human carry this many bone disorders in a way never recorded before or since? What kind of genetic inheritance or environmental stress could produce something so rare, yet so human? And if a skeleton like this could lie undiscovered in Chile's desert for decades, what else is buried there? Because not far from Atta's grave, another team unearthed something even more impossible. A disease they said was brought by Europeans. But the DNA told another story. A story 4,000 years older. Northern Chile. Archaeological teams dig beneath the sands of El Cerrito and La Herradura, chasing signs of early civilization. What they find are 2 human skeletons. Males. Adults. 1 over 40. The bones are strangely thickened, faces disfigured, nasal bridges collapsed like they'd been eaten away. At first glance, it looks like trauma, or maybe a genetic deformity. But then the DNA results come in. Trapped inside the jawbone, sealed for millennia by the Atacama's perfect dryness, is the genome of Mycobacterium lepromatosis. Leprosy. Not the medieval one. Not the kind whispered about in European history books. This one is different. Older. Deeper. 4,000 years old. And its genes hold a secret the world wasn't ready for. In a stunning paper led by international microbiologists, researchers discover 94 unique mutations in the ancient strain, mutations not seen in any modern leprosy cases. The analysis proves something shocking. This wasn't imported into the Americas. It originated here. The strain diverged from its cousin M. lepre over 26,800 years ago, likely evolving in isolation with the first humans who reached South America. It rewrites centuries of medical belief, because for 500 years, historians taught that leprosy came to the Americas on European ships, carried by conquistadors and colonists after 1492. But this strain was already here, in full force, by 2000 BC, which means indigenous societies were battling chronic, deforming disease long before Columbus was even born. It means they developed treatments, burial practices, maybe even spiritual beliefs around something we never imagined existed in their world. The bones tell us this wasn't a rare case. It was a community event, and likely a long-forgotten epidemic. But if leprosy didn't arrive from Europe, what else did first Americans bring with them? Because deep inside their DNA, researchers would soon find the traces of something far bigger. Not a disease this time, but an entire journey. The longest human migration ever recorded. It began not in Chile, but nearly 13,000 miles away. In West Beringia, now eastern Siberia, somewhere around 25,000 to 18,000 years ago, a population split from Northeast Asians. They waited, trapped by ice, in a place scientists now call the Beringian Standstill Zone. For thousands of years, they didn't move, they adapted, mutated, became something new. And then the glaciers cracked. Around 14,000 years ago, these early humans surged southward through the Americas, carrying more than just tools and fire. They brought their immune systems, their gut microbiomes, and their ancient pathogens. They crossed the Andes, cut through jungles, navigated rivers, and some kept walking until they reached the edge of the world. Patagonia, southern Chile, where the Pacific beats against black stone like a war drum. The trek, over 12,400 miles. No migration in human history has gone farther, on foot, in one direction. And now, using genome-wide data from 1,537 individuals across 139 ethnic groups, scientists have mapped it back together, piece by genetic piece. They found 4 distinct lineages across South America: Amazonian, Andean, Chicoan, and Patagonian, each one shaped by geography, isolated by mountains, deserts, ice fields, each one carrying fewer variations in their HLA genes, the immune system's radar. The deeper they traveled, the more their diversity thinned. It explains the tragedy of later centuries. Why smallpox and measles hit so hard. Why entire civilizations fell in weeks. They weren't weak, they were isolated. For thousands of years. Long enough for their DNA to forget how to fight what the old world would bring. But in all that data, scientists found one anomaly. In the far south of Chile, past the rivers, past the Andes, beyond where horses would ever go, a genetic lineage survived that didn't mix, didn't change. For 7,000 years, it stayed untouched. The question is how? And what kind of people could hold a genetic identity so strong it refused to disappear? To find the answer, we go south, farther than empires, beyond the frontiers of maps. Welcome to Patagonia, a windswept edge of the continent where glaciers groan, and the sea knows no silence. Here, DNA tells a story no conquest ever erased. Between 6,600 and 4,700 years before present, along the fjords and inlets of southern Chile, lived 3 distinct indigenous groups: the Coscar, the Yamana, and the Selknam. They did not farm. They fished. They did not build temples. They built canoes, crafted from bark and bone, passed from hand to hand like memory. And when researchers sequenced their remains, they discovered something extraordinary. No Eurasian markers, no African admixture, no blending from the north. Just pure, unbroken Native American lineage. Mitochondrial haplogroups C and D, passed from mother to daughter. Y chromosome haplogroup QM3, carried through fathers, stretching back to the first arrivals from Beringia. No contamination, even after 500 years of colonial pressure. In a world rewritten by ships and swords, these were the people who said no. Not with words, but with isolation. Even burial customs held their ground. Bodies laid curled in fetal position, wrapped in sea lion skin, a practice unchanged for thousands of years. In the coastal middens of Navarino Island, bones lie layered with shellfish and tools, evidence of maritime lifeways stretching back 7 millennia. Cultural fingerprints so stable, they might as well be carved in basalt. This wasn't genetic resilience born of resistance, it was geography, environment, and ancestral will working in harmony. But while the far south remained frozen in time, the center of the country did not. Because just above these untouched lineages, another story begins to rise. A story where the DNA didn't hold its shape. Because 2 worlds collided. And in that collision, something irreversible was written into the blood. They called it mestizage. But the genome tells it differently. In 2020, when Chilean genome-wide studies mapped the uniparental markers of thousands of individuals, the truth sharpened like a blade. 88% of mitochondrial DNA, the maternal line, was indigenous. 92% of Y chromosomes, the paternal line, were European. It was a pattern repeated across city and mountain, desert and coast. And it wasn't random. The Spanish arrived in the mid-1500s, led by Pedro de Valdivia, carrying steel, crosses, and royal charters. But they did not bring their women. From the start of colonization, indigenous women became the mothers of a new country, not by choice, but by conquest. The DNA proves it. Haplogroup A2, mostly found in the north, reflects Aymara and Atacamino roots. In the south, D1 dominates, an ancient marker tied to the Mapuche, the only indigenous group to resist full Spanish control for over 3 centuries. But even resistance has limits. By the late 1600s, mixed unions weren't just common, they were law. Royal decrees encouraged it. Catholic priests baptized it. What they called assimilation, the genome remembers as imbalance. Because while the Spanish names endured, Fernandez, Gonzalez, Rojas, the native women's names vanished, erased from the records, but not from the biology. Their blood carried the mitochondrial line forward, generation after generation. Quietly, invisibly, unbroken. This wasn't just cultural mixing. It was gender genetic rewriting. A blueprint of history passed from womb to child, while the father's name went to the church. But buried deep inside this asymmetric inheritance, something strange started appearing. Because not all Y-DNA was Spanish, and not all indigenous genomes came from Chile. Some came from across oceans, carrying whispers of Africa, of Asia, and even of a forgotten people from the eastern Mediterranean, whose story was never meant to reach these shores. But their story did reach, not in books, not in statues, but in blood. In 2023, Chilean ancestry databases began to show something strange. A pattern. Tiny, trace amounts of ancestry no one could explain. Less than 1%, but always there. Romani, Sephardic Jewish, North Indian, West African. In the capital, Santiago, once the center of colonial power, 17th century records reveal something shocking. Over 30% of the population was of African descent. By the 1700s, that number vanishes, not through migration, but through absorption, through erasure. Their names are gone from census rolls, but their DNA remains. Genetic tests now show up to 3.9% African ancestry in the north of Chile, dropping to about 1% in the south. Most of it from places like Senegal, Cameroon, Angola, brought during the 17th and 18th centuries, when enslaved Africans arrived at Chile's mining centers and ports. And then there's the Sephardic Trail. Around 1492, Spain expelled thousands of Jews. Some fled to Portugal. Some converted. Some, known as conversos, crossed the Atlantic. They changed names, buried customs, hid identities. But in modern Chilean DNA, scientists found a signature. 4% of sampled Chileans carry Sephardic Jewish ancestry. A ghost lineage hidden behind Spanish surnames, cloaked in Catholic rituals. And then the Romani. Their DNA, tied to northwest India, 0.8% in Chileans, tells of a journey spanning centuries, through Europe's shadows. They arrived with Spanish settlers, marginalized, forgotten, yet genetically present. These aren't fragments. They are threads, woven into the Chilean genome, quiet and unclaimed. Because when someone looks European, but carries the blood of India, of the Sahel, of Judea, what does identity even mean? Chile's genetic past isn't a straight line. It's a lattice of loss, migration, secrecy, and survival. But what if some of these migrations didn't follow land routes at all? What if they sailed? Because just off the Chilean coast, buried in bone and carbon, is a clue to a story even older. A transoceanic voyage science once said was impossible. Arauco Peninsula, 2022. Archaeologists brush away sand from a cluster of fragile chicken bones. Yellowed, brittle, forgotten. But when they test the mitochondrial DNA, it doesn't match Spanish breeds or European. It matches chickens from Polynesia. Specifically, the Lapita culture. Ancient navigators who launched out of Southeast Asia and fanned across the Pacific in outrigger canoes. And the radiocarbon dating? Pre-Columbian. Before Columbus. Before Magellan. Before any European knew this continent existed. For years, this theory had been mocked. A fringe idea whispered in Pacific anthropology circles. But now, the DNA said otherwise. It wasn't just chickens. the map starts shifting. In Chilean boat design, carved prow tips that mirror canoes from Fiji. In oral stories, Mapuche legends of men with darker skin, arriving from the west, vanishing with the tide. And in burial layers, carbon samples from Monte Verde, Valdivia, and coastal midden sites, all suggesting contact that defies the timelines written in textbooks. If Polynesians really reached South America, it means they crossed 7,000 kilometers of open ocean. No compass, no maps, just stars, wind, memory. And if they brought chickens, what else did they bring? Language, crops, genes. Studies are now re-examining skull shapes, textile patterns, and, most of all, human DNA, looking for traces of Polynesian ancestry in Chilean lineages. The early results? Inconclusive. But something is there. Something that shouldn't be. Because if Pacific voyagers reached South America centuries before Europe ever did, then the story of global contact isn't about 1492. It's about an ancient ocean, and the people who dared to cross it first. But it wasn't only voyagers who reached Chile by sea. Sometimes, what washed ashore was sickness, and one forgotten disease, trapped in Chilean bones for 4,000 years, would shatter everything we thought we knew about who got sick first, and why. Because leprosy wasn't alone. In the bone-dry depths of the Atacama Desert, scientists didn't just find skeletons. They found a war, fought in silence, under the skin, centuries before recorded history. In rodent middens, hardened nests of fossilized feces, and deep burial shafts, researchers uncovered something shocking. Ancient microbial DNA, so well preserved, it still carried the genetic fingerprints of infection. And among it, tuberculosis, actinobacteria, and unidentified strains that don't exist anywhere in modern medicine. One sample, extracted from a child's rib near San Pedro de Atacama, showed lesions typical of pulmonary TB. The DNA confirmed it, Mycobacterium tuberculosis complex, present 1,000 years before European contact. This flips the old theory on its head. TB didn't come with the conquistadors. It was already here, spreading quietly through pre-Columbian villages, long before ships ever crossed the Atlantic. And it wasn't just TB. New research, funded by the Chilean Chancellor's Grant for Paleopathogen Studies, mapped DNA from soil layers dating back over 50,000 years. That data revealed dramatic microbial shifts, coinciding with climate events, glacier retreats, and human migration waves. These pathogens weren't just infections, they were evolutionary agents. They adapted as humans moved. They mutated with ecosystems. And sometimes they killed. But now, scientists are using those same ancient genomes to predict the next wave. By studying how past pandemics spread through isolated populations, they're building models for modern zoonotic threats. The kind that leap from animals to humans. The kind that still start in dry corners of the earth. Because in Atacama, the bones whisper warnings. Epidemics don't need cities. They only need breath, blood, and time. But while the desert holds the memory of disease, the living still carry something stranger. A genetic fusion, born of every migration, every storm, every collision. And in the genome of the modern Chilean, that fusion has a shape, a pattern, a code. But what happens when your body carries bloodlines that history never recorded? What does it mean to look like one thing and be made of 1,000 stories? Because in modern Chile, identity isn't a mirror. It's a mosaic. In 2020, a sweeping genome-wide study shook the foundation of what it meant to be Chilean. When researchers sequenced thousands of individuals across all regions, they didn't find one ancestry. They found layers. The average genome? 42% Native American, primarily Aymara, Atacamino Mapuche. 55% European, mostly Iberian, but also Italian, Basque, and Balkan. 2 to 3% Sub-Saharan African, the quiet echo of enslaved lives long erased from textbooks. But this isn't evenly spread. In the north, Aymara genes dominate. In the center, near Santiago, European markers rise. And in the deep south, Patagonia, Punta Arenas, the contradiction becomes stark. There, 84% of mitochondrial DNA, the mother line, is native. But 92% of Y chromosomes, the father line, are European. It reflects a newer wave of migration, Croatian, German, and British settlers, arriving after 1845, shaping the southern genome like a chisel-shaped stone. So what does someone from Chile look like? It depends. On altitude, on coastlines, on conquest, on who your grandmother was, and what your grandfather's passport said. And even that is no longer enough. In 2023, Chilean researchers conducted a massive GWAS, a genome-wide association study, focused on Alzheimer's risk. They discovered something alarming. European risk models failed when applied to Chilean genomes. Without local ancestry calibration, predictions were inaccurate and dangerous. Because the Chilean genome isn't Spanish. It isn't Mapuche. It isn't African, Jewish, Romani, or Polynesian. It's all of them, written in fragments, scattered like seeds. So now, the question isn't who are you? The question is, what ancient roads built your body? Because when we say Chilean, are we talking about language, location, or a 10,000-year-old map etched in blood? And if that map keeps unfolding, what happens when your DNA tells a story that even you didn't know was yours? Some of those stories aren't even finished. And one began with a skeleton the size of a doll. She was called Ada. Found in 2003, buried beneath a church ruin in La Noria, in the heart of the Atacama. 6 inches long, 10 ribs, a skull that didn't look human, and yet she was. Not an alien, not a hoax, but a human girl, a fetus, with mutations that science had never seen before. In 2018, Stanford's Dr. Gary Nolan published the full genomic sequencing. The results sent shockwaves. Ada carried 7 severe mutations, including 4 that were completely novel to science, affecting bone growth, cartilage formation, and cell signaling. One gene, COL1A1, linked her to brittle bone disease, but the mutation was unique. This was not a known condition. It was something new. Now, researchers across the world, including Dr. Kirsten Boss from the Max Planck Institute, are asking a new question. How many more Adas are out there? Because Chile isn't just a country. It's a vault. The Atacama Desert, with its ultra-arid climate, near-zero rainfall, and salt-enriched soils, has preserved genomes, bacteria, viruses, and pathogens for over 50,000 years. Rodent middens have trapped airborne disease particles. Soil samples carry RNA fragments from prehistoric microbes. Even lice found in mummified hair still hold blood traces from hosts long gone. This is no longer anthropology. This is bioarchaeological time travel. Every genome pulled from this land rewrites another rule about disease, migration, extinction, and memory. And these aren't just ancient clues. They're alive in modern immune systems, in vulnerability markers, in gene expressions still triggered by stress and environment. Ada wasn't an ending. She was a beginning. And somewhere, under layers of nitrate, under centuries of wind-buried silence, another skeleton waits. But this time, it won't have any known haplogroup, no ancestral match, no phylogenetic anchor. So what happens when the next Atacama skeleton is unearthed, and it doesn't fit anywhere on the human tree? What if it's something else entirely?